Okay, we now have gone from the second line to the third line. The first line, remember, were barriers. The second line was if you get underneath the epidermal area, let's say, and that was where you had phagocytosis, natural killer cells, fever, complement, interferons, and everything. We're to the third line. The third line is one that has to be created. The others two, which were the innate, it will fight against anything. But now in this third line, it has to be created. So there's a lag time. And this is called the adaptive defenses, the third line. After you get past that, you're going to need help, outside help. So it protects against infectious agents, abnormal body cells, which would mean like cancer cells, amplifies the inflammatory response, and you can activate complement. All right, here we go. The three things I want you to keep in mind on the third line is it is specific, very specific. It has to be cut and created. It is systemic. What does that mean? With the second line, if you get a bee sting on your hand, inflammation, it will only be on the hand. But when you activate the third line, it goes all over the body. So it protects all over the body, has memory. With the first and the second line, if you get attacked by a microorganism, it'll fight it, let's say, but it'll never remember it again. So you have no protection you'll have to start all over. With the adaptive, it memorizes the situation. So when it comes again, it's ready to go. It doesn't need any lag time or anything. So the three things about the third line is specific for the certain antigen. It goes all over the body, systemic, and it has memory. Now, the two parts of the third line are called the humoral immunity and cell-mediated. Humoral immunity, humor is like a vapor. That means the humoral immunity is antibodies, and it kills stuff in the extracellular fluid. Now let me say it again. The humoral is to fight organisms in the extracellular fluid, outside the cell. The cell-mediated immunity fights organisms inside a cell, which means you have to take the whole cell out. So the humoral fights outside and the cell-mediated fights inside, but it has to take the whole cell out. The humoral immunity is going to be based upon B lymphocytes, B, like in boy, lymphocytes, we'll talk about. And the cellular immunity is based on T lymphocytes that we'll talk about. But both of them work together. So please read on this. Let's hit some terms. An antigen is a substance that can be recognized by that third line of defense. That's an antigen. It's a foreign substance. It can be a partial or complete antigen we're talking about. It can be a partial or complete. And we'll come to that in a minute. An isoantigen. Now, this makes me introduce something here called the MHC, major histocompatibility complex. So isoantigen versus heteroantigen. All right. On your cells, you have flags. Remember those CDs? On my cells, I have flags. But my flags, when it comes to immune action, are different than your flags. Because I have flags on my cells that say I'm me. You have set flags on your cells that say you're you. Now these flags actually are antigens. But early in your development, which we'll talk about, you develop tolerance. You develop tolerance so you wouldn't fight yourself, autoimmunity. You develop tolerance so you wouldn't fight yourself. So if I take one of your cells and put it in me, though you did not attack your flag, I would. So in you, it's an isoantigen. Why do we say an antigen? Because it would stimulate the human immune response. It's not stimulating it in you because you develop tolerance. But it is an antigen. So in you, it's your isoantigen. But if you put that cell in me, it's a heteroantigen because my body will fight it. So we have isoantigens and hetero. Then we're going to get to the MHC in a little while. So we have isoantigens and hetero, but they are antigens. An immunogen means a generator of the immune system. Is a special type of antigen that can stimulate the third line. Now, when I say stimulate the third line, that means make it create fighters. Make it create fighters. That's what we call an immunogen. It will make the third line create fighters. So we'll call it a complete antigen, and I'll explain that in a moment. So we have antigen, isoantigen, heteroantigen, immunogen, which is a complete antigen. Okay? Now, most antigens, when we look at it, are complex organic molecules not found in the body. Proteins are generally the most antigenic because they have the most diversity. Proteins are very antigenic because they have a considerable amount of diversity. Okay? Now, a complete antigen, immunogenicity. All right, this is what I want you to think about. Let's say we had an ink pen that had a cap on it. An ink pen that had a cap on it. Okay? Once you get past the first line and second line, if something gets to the third line won't create fighters to something unless it has enough relevance. The third line will not create fighters unless it has enough relevance. The relevance generally has to do with the size of the molecules. Large molecules, it generally will do something against. Small ones, it may not want it there, but it says it's a non-factor, it's insignificant. So it'll just let it ride it out. Okay, so if, let's say you had the ink pen with the cap, and we just use it theoretical. If it got into the third line, let's say that is big enough to stimulate the third line to create fighters. Thus it has immunogenicity. It's an immunogen. Immunogenicity, ability to stimulate proliferation of lymphocytes. In other words, it will cause it to fight, to create fighters. It's big enough, once the fighters are created, then the fighters will go after the thing. That means reactivity. Create the fighters and then go after it. So immunogenicity and reactivity. A complete antigen, because of its molecular size, has both create the fighters and go after it. Okay? And so now we come to, so that's a complete. Now I come to a partial antigen, also called an incomplete antigen, also called a hapten. They generally are small molecules, less than 1,000 atomic mass units, not immunogenic by themselves. Okay, let's just go back to our scenario. So the ink pen with the cap on it and the other part got in. It was large enough to cause stimulation and reactivity, immunogenicity and reactivity. All right, now this is what could happen. Let's say something happened that just the cap of the ink pen got in the next time, just using this as a scenario. Now, the cap of the ink pen would not have been big enough by itself to cause the third line to do anything. But because the larger entity had already gotten in and you had created fighters against the whole thing, then if the cap came in by itself, you would fight against it because you'd already created fighters to the whole thing. Thus, the cap would have been a partial antigen. The cap by itself, if it had gotten in, would not have done anything in the third line. But because you'd already done something in the third line, then the cap gets in and you fight against it. So thus, a partial antigen has reactivity. You'll fight against it, but not immunogenicity. Reactivity, but not immunogenicity. So kind of read on that if you would to get an understanding. Now, let's say a bacteria gets in. You really aren't, the third line is really not looking at the whole bacteria. What it's looking at are certain parts on the surface of that bacteria that we call antigenic determinants or epitopes. So the offender is really on the surface. The offender is really the surface that it's looking at. Now, once it sees the offender, it'll take out the whole thing. But these antigenic determinants, which are on the surface, are really the thing that the third line players look at, third line cells, the antigenic determinants. All right. Now, the most naturally occurring antigens have numerous antigenic determinants, like a bacteria or so forth. Now let's look at what I'm talking about. Take this for, let's say this is a bacteria, and let's say all of these are antigenic determinants that the cells would look at. The cells would look at. Okay. So most have several antigenic determinants. Many of these are diversified. So these here would be different than these here. Now what happens along the way in your immune system is that your parents handed on to you. We're going to use this scenario. Certain skeleton key third line cells based upon what they saw in their lifetime and their grandparents. So you have certain skeleton key fits already that you would work from. So if an organism got into you and your parents didn't hand you this quote skeleton key fit, you would have nothing to cut and snip from to make an antibody. You would have nothing to cut and snip from to make an antibody. But if something got in and it had a bunch of different antigenic determinants, then more than likely your parents would have passed you on a skeleton key that could fit some of these. So the more diverse the antigenic determinants, the more likely you have in your genes, your DNA, recognition of something that can fight it. So that's the reason that in the third line, you really don't fight against plastics or metals. That's why, a prosthesis is like artificial. That's why artificial things that are plastic or metal, they wouldn't have any antigenic determinants. And if they did, they would be uniform. What parent is going to hand you in the genetics, a fight against a plastic or a metal? So that's why prostheses don't really cause anything. Now, mind you, we put a prosthesis in, it will cause inflammation, but it won't activate that third line. It won't activate that third line unless something has fallen on it that you've already, like a hapten or something. Okay? So antigenic determinants on there, and the more diverse, that's why you see here, here's an antigen, and see these antigenic determinants, the more diverse, the more likelihood you would have something to recognize some of these. The more uniform, the more likelihood you won't in that third line. So read through your book on that. I know it's hard to explain without being face to face. Perhaps maybe when I come to the college for a 2-hour session, you can ask a question on that or email me or something. All right. So we go here again to isoantigens, which are not being attacked in you, but they are antigens. They're your self-antigens, the isoantigens. So we're going to start here in a moment. I'm going to end this video.